Hello students, how are you doing? Welcome to another biology class. Today we will learn about chroloplast, and this is the first part of the lesson. Before we start, make sure you have subscribed to this channel. Alright then, let us begin. Chloroplast. A chloroplast is a type of membrane-bound organelle known as a plastid that conducts photosynthesis mostly in plant and algal cells. The photosynthetic pigment chlorophyll captures the energy from sunlight, converts it, and stores it in the energy storage molecules ATP and MTF while freeing oxygen from water in the cells. The ATP and MTF is then used to make organic molecules from carbon dioxide in a process known as the Calvin cycle. Chloroplasts carry out a number of other functions, including fatty acid synthesis, amino acid synthesis, and the immune response in plants. The number of chloroplasts per cell varies from 1 in unicellular algae up to 100 in plants like Arabidopsis and wheat. A chloroplast is characterized by its two membranes and a high concentration of chlorophyll. Other plastid types, such as the leucoplast and the chromoplast, contain little chlorophyll and do not carry out photosynthesis. Chloroplasts are highly dynamic. They circulate and are moved around within plant cells, and occasionally pinch into to reproduce.
Their behavior is strongly influenced by environmental factors like light, color, and intensity. Chloroplasts, like mitochondria, contain their own DNA, which is thought to be inherited from their ancestor, a photosynthetic cyanobacterium that was engulfed by an early eukaryotic cell. Chloroplasts cannot be made by the plant cell and must be inherited by each daughter cell during cell division. With one exception, the amoeboid Polynella chromatophora, all chloroplasts can probably be traced back to a single endosymbiotic event, when a cyanobacterium was engulfed by the eukaryote. Despite this, chloroplasts can be found in an extremely wide set of organisms, some not even directly related to each other, a consequence of many secondary and even tertiary endosymbiotic events. The word chloroplast is derived from the Greek words chloros, which means green, and plasts, which means the one who forms. Discover the first definitive description of a chloroplast, chlorophyll cornon, grain of chlorophyll, was given by Hugo von Mohl in 1837 as discrete bodies within the green plant cell. In 1883, Andreas Franz Wilhelm Schimper would name these bodies as chloroplastids, chloroplastiden. In 1884, Edward Strasburger adopted the term chloroplasts, chloroplastin. Lineages and evolution. Chloroplasts are one of many types of organelles in the plant cell.
They are considered to have evolved from endosymbiotic cyanobacteria. Mitochondria are thought to have come from a similar endosymbiosis event, where an aerobic prokaryote was engulfed. This origin of chloroplasts was first suggested by the Russian biologist Konstantin Merezhkovsky in 1905 after Andreas Franz Wilhelm Schimper observed in 1883 that chloroplasts closely resemble cyanobacteria. Chloroplasts are only found in plants, algae, and 3 species of amoeba: Polynella chromatophora, P. micropora, and marine P. longichromatophorae. Parent group: Cyanobacteria. Chloroplasts are considered endosymbiotic. Cyanobacteria. Cyanobacteria are sometimes called blue-green algae even though they are prokaryotes. They are a diverse phylum of gram-negative bacteria capable of carrying out photosynthesis. Cyanobacteria also contain a peptidoglycan cell wall, which is thicker than another gram-negative bacteria, and which is located between their 2 cell membranes. Like chloroplasts, they have thylakoids within them. On the thylakoid membranes are photosynthetic pigments, including chlorophyll A. Phycobilins are also common cyanobacterial pigments, usually organized into hemispherical phycobilisms attached to the outside of the thylakoid membranes. Phycobilins are not shared with all chloroplasts though.
Primary endosymbiosis. A eukaryote with mitochondria engulfed a cyanobacterium in an event of serial primary endosymbiosis, creating a lineage of cells with both organelles. It is important to note that the cyanobacterial endosymbiont already had a double membrane. The phagosomal vacuole-derived membrane was lost. Primary endosymbiosis. A eukaryote with mitochondria engulfed a cyanobacterium in an event of serial primary endosymbiosis, creating a lineage of cells with both organelles. It is important to note that the cyanobacterial endosymbiont already had a double membrane. The phagosomal vacuole-derived membrane was lost. Somewhere between 1 and 2 billion years ago, A free-living cyanobacterium entered an early eukaryotic cell, either as food or as an internal parasite, but managed to escape the phagocytic vacuole it was contained in. The two innermost lipid bilayer membranes that surround all chloroplasts correspond to the outer and inner membranes of the ancestral cyanobacterium's gram-negative cell wall, and not the phagosomal membrane from the host, which was probably lost.
The new cellular resident quickly became an advantage, providing food for the eukaryotic host, which allowed it to live within it. Over time, the cyanobacterium was assimilated, and many of its genes were lost or transferred to the nucleus of the host. From genomes that probably originally contained over 3,000 genes, only about 130 genes remain in the chloroplasts of contemporary plants. Some of its proteins were then synthesized in the cytoplasm of the host cell, and imported back into the chloroplast, formerly the cyanobacterium. Separately, somewhere about 90-140 million years ago, it happened again and led to the amoeboid Polynella chromatophora. This event is called endosymbiosis, or cell living inside another cell with a mutual benefit for both. The external cell is commonly referred to as the host, while the internal cell is called the endosymbiont. Chloroplasts are believed to have arisen after mitochondria, since all eukaryotes contain mitochondria, but not all have chloroplasts. This is called serial endosymbiosis, an early eukaryote engulfing the mitochondrion ancestor, and some descendants of it then engulfing the chloroplast ancestor, creating a cell with both chloroplasts and mitochondria. Whether or not primary chloroplasts came from a single endosymbiotic event, or many independent engulfments across various eukaryotic lineages, has long been debated. It is now generally held that organisms with primary chloroplasts share a single ancestor that took in a cyanobacterium 600-2000 million years ago. It has been proposed the closest living relative of this bacterium is Gloeomargarita lithifera. The exception is the amoeboid Polynella chromatophora, which descends from an ancestor that took in a Prochlorococcus cyanobacterium 90-500 million years ago. These chloroplasts, which can be traced back directly to a cyanobacterial ancestor, are known as primary plastids. Plastid in this context means almost the same thing as chloroplast. All primary chloroplasts belong to one of four chloroplast lineages: the glaucophyte chloroplast lineage, the amoeboid Polynella chromatophora lineage, the rhodophyte red algal chloroplast lineage, or the chloroplastidin green chloroplast lineage. The rhodophyte and chloroplastidin lineages are the largest, with chloroplastidin green being the one that contains the land plan.
That is all for today, folks. One last thing. Before you go, make sure that you have subscribed to our YouTube channel and be kind to like and share this video. This video is edited by Baruch Solomon, MSc in Genetics. I will see you in the next video. Goodbye.